Western blotting, also known as immunoblotting, is a crucial technique used in molecular biology to detect specific proteins from a complex mixture. It's widely used in research for studying protein expression, modifications, and interactions. Let's walk through the key steps. First, sample preparation. Proteins are extracted from cells or tissues using special lysis buffers that break open the cells. Protease inhibitors are added to prevent the breakdown of proteins during the process. Then, we measure the protein concentration and load equal amounts onto a gel for accurate comparisons between samples. Next is gel electrophoresis, specifically SDS page. Here, sodium dodecyl sulfate, or SDS, coats the proteins, giving them a uniform negative charge. When we apply an electric current, the proteins are separated based on their size. Smaller proteins move faster, while larger ones move more slowly. After separation, proteins form distinct bands within the gel. In the transfer step, the proteins are transferred onto a membrane, usually made of nitrocellulose or PVDF. This is done through electroblotting, where an electric current pulls the proteins from the gel onto the membrane. Now, the membrane has an imprint of the proteins in their separated positions, ready for detection. To prevent nonspecific binding during detection, the membrane is blocked with a solution containing proteins like bovine serum albumin. This ensures that the antibodies used later bind only to the target protein and not the rest of the membrane. Now comes antibody incubation. The membrane is incubated with a primary antibody that specifically recognizes the protein of interest. After washing off unbound antibodies, we add a secondary antibody that binds to the primary one. This secondary antibody is linked to a detection molecule, like an enzyme or fluorescent dye, which will help us visualize the protein. Finally, we move to detection. There are a few common methods. For example, in chemiluminescence, the secondary antibody reacts with a substrate, producing light. The intensity of this signal corresponds to the amount of the target protein, allowing us to analyze its expression. Now, let's discuss the applications. Western blotting is widely used to measure protein expression under different conditions, like during disease or drug treatments. It can also detect post-translational modifications, such as phosphorylation or glycosylation, which affect protein function. Clinically, it's a tool in diagnosing diseases, including HIV and prion diseases. Western blotting offers several advantages. It's highly specific, thanks to antibody detection, and can provide semi-quantitative information on protein levels. Plus, it's versatile, working with various biological samples like cell lysates and tissue extracts. However, it's not without its challenges. The process can be time-consuming and relies heavily on the quality of the antibodies used. Additionally, it's only semi-quantitative, and detecting proteins in extremely high or low amounts can be tricky. In conclusion, Western blotting remains a powerful and reliable method for protein analysis. Its ability to separate and detect proteins with precision continues to make it invaluable in both research and clinical diagnostics.